So we're focused on this translation part of it today and how we take this messenger RNA and how that spits out a protein. So if you guys remember, there is this little chart here. Now, I gave everyone should have this document, right? I will load that up for those of you online. Everyone should have this document. There's a couple things here. Like there's some wheels and charts. Online, you guys should have this too. If you don't, it is on the LMS. The key things we're kind of focused on here are these bottom two. I prefer the tables. Some people prefer the wheels, and I will teach you how to read this shortly. So we will end up using this a lot today. So we will be referencing this sheet a lot. But basically, if you guys remember, these codons here, these are called the codons. They're read 3 at a time. So these things are read 3 at a time by the ribosome. And what's going to happen is it's going to spit it in, and it's going to go into the ribosome here, and we're going to see that, here we go. It's going to go into the ribosome here, and then there's going to be something called an anticodon, which is going to be just a complementary pair. It's going to send that into the ribosome, and it's going to give you back a protein. So the mRNA comes in, it gets read, we get an anticodon, and then it's going to spit out a protein. So that is where we're focused on today. So this is the new stuff here. So there will not be a whole bunch more notes, to be honest with you, with this section. A lot of this is going to be us doing this. So ribosomes specifically, though, the ribosomes are where these proteins get synthesized, created. So when the mRNA leaves the nucleus, you know what? You don't even need this part. Don't even need this part. You just need the top two. Ribosomes are where these codons get put into, where the mRNA goes to, and it's going to read these 3 at a time and spit out a protein. You don't need the middle two, just the top and the bottom part. And before we get going, I do just want to reiterate, there is no school on Monday. That does not exist. Reminder, we do have our final on the 30th. Online people, I need to know where you want to write your final. It has to be in person. It has to be at a Carleton site. It can be in Wadena, Humboldt, Waka, etc. I need to know where you want to write that. And you don't need to tell me right now, but if you want to, that's fine too. Okay, Jordan, you'll be in Humboldt. Okay. Paige, I assume you're going to write here in Wadena. Okay, perfect. Awesome. Okay. Is everyone done writing? So what we're going to do is we're going to go through the process of how do you actually read this stuff? How do I go from how is this translated and how does this spit out a protein? So that's what we're focused on too. And there's a little bit of a science behind it. So that's where we're going to end up going into next. Don't need this. This is useless.
When are we writing the exams? Sorry, I missed that part.
Oh, sorry. The exams, that's for the departmental.
Yeah, when is it? Like, when are we writing it?
It's on the 30th. Let me just confirm if it was the morning or the afternoon again. Exam schedule.
Because we have grad to attend in
Oh, sorry, the 29th. I meant the 29th. I apologize. 29th.
Oh, okay.
Yes. Let me just load up final exam schedule. Here we go. Because I forget which one's in the morning, which is So so the biology is in the afternoon. So just the same time as we normally do. Sorry, not same time. It's at 12:30. So you can come here from 12:30 and you will have until 3:30. I recommend you guys do come a bit early. I wanted to give you guys as much time to write as possible. And reminder for the final, like there's no breaks and stuff, like you got to just power through it. If you do need to say use the washroom or something, you got to leave everything on your table, including your like phone and stuff. You can't go outside. So the biology is on the afternoon of the 29th. Yeah, thank you for catching me on that. Okay, so we're gonna watch part of a video here. This guy does a really good job of explaining it. So I'm just gonna let him do it here. We're not gonna watch the full thing, but we're gonna look at this translation process. And then we're gonna eventually end up doing this. So we're gonna learn how we can translate stuff. If I give you a bunch of DNA codes such as, here we go.
So when
if I give you DNA, if I give you this, we're going to learn how to turn this into mRNA, into tRNA, into a protein. That's what we're doing now. This guy does a really good job of it, but I will also walk you through it. Okay, so I'm gonna watch this for a bit.
So when we look at the process of translation, this is the actual process where the ribosome will create a protein. And first of all, here comes our messenger RNA. It's arrived at the ribosome. Notice that transcription has just ended. And what happens next is the ribosome will read the codons once it encounters AUG. And I've highlighted AUG right there in red. So right now, nothing's really happening. The ribosome hasn't come to AUG yet. But now it has. Now the ribosome's at AUG. And so the ribosome, the AUG will turn on the ribosome and start the sequence. And then transfer RNA will deliver the proper amino acid. And we use this chart right here to figure out what's the proper amino acid. We look up AUG. AUG on the chart, yes, it's a start codon, but it's also, notice to the right of AUG, is the word methionine. Transfer RNA should deliver methionine. And in my animation, here comes this weirdly shaped object. That's transfer RNA. It's carrying an orange circle named methionine. But how does the ribosome know that this is the right amino acid? Turns out there's a hidden secret on tRNA. tRNA actually has 3 nucleotides itself called the anticodon. They're going to drop out of the tRNA right now. Watch what happens. Watch the tRNA. 3, 2, 1. On tRNA are 3 nucleotides as well. Notice this is what we call the anticodon. Anti is a prefix that means opposite. Anticodon means opposite the codon. UAC, that's opposite of AUG because they are all 3 accurate opposites. This is the safety method that prevents the delivery of the wrong amino acid. The anticodon and the codon match. And because they match, that is the chemical signal that we have the proper amino acid. So now the ribosome will move to the next codon, highlighted red CCA. The ribosome is moved to the next codon. Well, we get out the chart. We find CCA on the chart. And
so again, so just kind of reiterate what happened here. Again, we won't go through the whole video, but again, so here's all my DNA encoded, right? So I've taken my DNA. This is the mRNA now, messenger RNA. It gets into that ribosome, and it's just going to read it. It's just going to chill until it sees this thing called AUG. This is the only codon. You need to have those three in a row that this tells it to turn on. Now it's going to read it. It's going to start producing proteins 3 at a time. So it goes AUG, CCA, then GGA, and UCG, and so on and so forth. And so, as a way to spell check it as well, it's going to deliver whatever protein it is, and we will learn how to find that on here. It's going to deliver whatever protein there is on here, and then it's going to use an anticodon, the opposite, to make sure that match. Think about a lock and a key. It's making sure that that matches. If it unlocks, here's your protein. If the key doesn't fit, if the anticodon is incorrect, that means, oh crap, I made the wrong protein. I'm going to get rid of this now. So that's what it does. I don't think we got to go through all of this here. No, we don't. He just does it like step by step by step by step by step. That's what we're going to end up doing here. Okay. So we are going to do this. So we are going to create, turn this into mRNA and then tRNA. In fact, let me just quickly, I want to edit, I want to edit this a little bit. I'd like formatting it differently now. Here we go. So here's my DNA. We're going to turn this into mRNA, which is uncapitalized correctly. We're going to turn that into tRNA, and then we're going to turn that into an amino, we'll say protein, technically an amino acid, same thing. Here we go. So you can put, write this into your book here, and we will go through all the steps of this. So I goofed up on the recording here, but just as a quick summary, I took my DNA, I put it in the batches of 3, and then I took that and I created the mRNA that I just went the opposite of. Instead of T's, it's an A. Instead of an A, it's supposed to be a T, but for RNA, it's a U. And then for C, the opposite of that's a G, and I just kept doing that throughout. Then I repeated that process from mRNA to our anticodon tRNA. So now I did UAC, and then I did CGCU is CGA, et cetera, et cetera. And now my last step here is I'm going to create a protein out of this. Now, this is a really common mistake. This tRNA, what is that? It is the spell check. So when we see tRNA, we use that as a spell check. The protein is based off the mRNA. So it's really important. You are not using the tRNA to generate it. You are using the mRNA to do it. So now what I'm going to do is I'm going to use that chart, that wheel, those wheels or charts from before. You've already seen a couple examples of them. Sometimes it's a chart, sometimes it's a wheel. There's 1,000 different of them. The one thing I want to note is you do not need to memorize it. You do not need to memorize that chart. But you need to know which codon starts the process. And so the codon that starts the process is this one here, AUG. AUG, methionine, starts it. Nothing happens until your ribosome reads AUG, and then it starts. And you guys can see here, the first thing I found off my mRNA, not my tRNA, is AUG. So let's start spitting that out here now. So instead of AUG, I'm going to put what that protein is. So what I do is I go to the first base of it, the first letter, A. Then I go to wherever it is. In this case, it's U. But if it was AAC, for example, it would be like I'd use this as my second one, and then I look at my third one. So you start at the left, you work your way up to the second letter, and you make your way right to the third letter. So AUG is A, U, and then G. It is methionine. You do not need to write that whole thing. You can instead shorthand it. Notice how it says MET. You would just put MET. That is shorthand for methionine. Next, I'm going to do my next one here. I'm going to look for GCU. I'll give you a second. Try and find that on your chart. You're going to try and find GCU on your chart. Give you 30 seconds to try and locate which protein that is. So the protein that is again, my first letter in this was G. I'm going to go to the G section down here. My second letter was, did I say it was C? So I know it's going to be in this square here. And then my third letter was U, which is this one here. So it's this one right here that I'm circling with my mouse. Now you're going to notice it doesn't have something right beside it. That's because all four of these are alanine. So in this case, it's going to be ALA. It's going to be alanine. I don't need to write alanine. I just need to write ALA. We do it shorthand. And I'm going to repeat that process here for everything. Next, AAU. AAU. And then U is ASN, asparaginine, ASN. Then I'm looking at a UGU. UGU. CYS, it's cysteine. And then I go for UGA. So U, G, and A, serenine, SER. So the one that's in the black box here is the protein that gets spit out. And again, the most common mistake people will make here is they will either they will start it before they see this AUG, or two, they will use the tRNA to code it. The tRNA is the spell check only. Now, there's one issue with this, which is technically this is an incomplete code. This DNA here is incomplete. There will continue to be proteins that spit out until I run into one of these three stops here on this top right, one of these three stops. Just like I have a green light, a start button of AUG, I have a red light, a stop button of UAA, UAG, and UGA. It will keep going until it spits that out. And once it sees one of those three, it's going to stop creating proteins. It's still going to read it. It's just going to ignore everything after it. So is there any questions on this before we go? It is important we know this. I know it's complicated. Mm-hmm. Yes, yes.
And then the second, the last one.
That's exactly it. So if I'm doing, let's say I'm doing ACG, I would find A, then I would find G, so I know it's right here. And then what did I say? ACG, AGC or something? Maybe that. Is that done?
Yeah.
Okay, yeah. Now, there is the chart You could also use, if you wanted, the wheel. People have different preferences. The wheel is you just work your way in and then work your way out, right? If I'm doing GUC, here's my G. Then it's U. And then it doesn't matter which one of those four it is, it's always going to be vowel line. But what if I do AGA? Sorry. Wait, what did I do here? No, okay, ignore that. Sorry, I confused myself there. Okay. So this is something we're going to be doing a little bit, a couple of times today. Don't need this video anymore. Come on. So this is what it would be, ultimately. Start alanine. If I were to do, if I were to create it, eventually I'd get start alanine, asparaginine, and cysteine with a different one. So what we're going to do here is we're going to finish this topic up, and then we're going to practice this skill a little bit more. Come on. So I got 4 questions here that we're going to do. Okay, you're going to need, you just need like 2 lines. I'm going to say like 2 for each of these, so 6 in total. You just need 1 line for this one. And then there's going to be a lot for number 4. I will give you 2 minutes just to get that stuff down. So starting out. Again, I'll give you a second here. See if you can kind of piece together which of these 4 things goes where. You don't have to write it down, but just think about it. Give you 20 seconds. Okay, so the first thing is, so RNA is created from the template strand of DNA. We create RNA from DNA. So DNA is the template, right? And then we're going to use RNA to create amino acids, which turn into proteins. Sorry. Okay, so next part here. We want to look at like what do each of these do? Again, there's so many RNAs. It's a really easy thing to get confused about is what the heck do I do for all of these? We have rRNA, which is a component of ribosome. We have rRNA, so major component. We have mRNA, so this carries that complementary code from that template strand, and this is what goes into the ribosome to create proteins. And tRNA is what transports those amino acids to create those proteins. And they also use the anticodon. Okay. And finally, how does DNA turn to proteins? The big thing here, there's a lot of stuff here before you start writing. The big thing you have to remember is the stuff on the left. DNA goes through transcription to become RNA, and then RNA goes through translation to become proteins. That's it in a nutshell. Obviously, though, it's more complicated, right? But if you remember this stuff on the left and you know what all 5 of these things are, you're good to go. And let's try and no, actually, I kind of don't. I really don't want to remove anything up there. If you are confident you know this, you don't have to write it down. But in a nutshell, this is what it is. All of this stuff here. Your DNA gets broken apart by that RNA polymerase. You get that. It creates an mRNA. It detaches, moves to a ribosome where it goes through transcription. And then it goes into the it goes into the cytoplasm ribosome, which uses translation. It reads it 3 at a time, it delivers the amino acid, and then it creates the protein. And we start it with AUG and we stop it with either one of these 3. All 3 of them do the same job. And that tRNA is that spell checker, which is that anticodon. That makes sure it's the correct match. So I know there's a lot there. If you are confident, you do not need to write it down. We're only 2 weeks into the final, so I'm confident you guys will remember it if you're confident now. Okay, so that's it for like the notes of this. We might start the next section later. I'm not sure. We will see. I want to give you time for something. Really quickly though, what I want you guys to try and do is just kind of a practice point here. I want you to try and fully translate this on your own. I will give you a hint that I'll just split it up. No, I won't. Actually, yes, I will. I'll just split it up there for you into 3 bases. Give you guys like 3 or 4 minutes to try that out. You're going to be expected to kind of do it like reverse style as well. So what I've done is I have created, I want to make it a little bit bigger. Perfect. So I'm going to hand this out in just a second. This is just an example of this. Then the first few are going to be the exact same way that we've done stuff. But it gets complicated. What if I give you a strand of mRNA or a partial strand of mRNA and I say figure out the rest of it? So you could, so what if it's just mRNA? Now we'll work backwards to get DNA, for example. So if I go from U's, well, what gives me a U when I go up here? It's an A. What gives me an A when I go up here? It's a T. Not a U, it's a T because I'm going to DNA, for example. What if I give you something that's a little bit of both and you have to work your way backwards? So what I'm going to give you guys here is some time to do this. I will hand this out here in just one quick second. But this is a good opportunity to practice it and get more difficult as we go. So this is for marks. I am pretty sure I printed this out for you guys online. If I didn't, I apologize. It is on the LMS. But this one is going to be, it is an assignment. And the whole goal of this is just some practice, right? I want to make sure we have this skill down because this is kind of like the culmination of the last 2 weeks of learning is this. So you might get the rest of the day for it. I'm not sure yet. I'll kind of wait and see how we're going. If you get stuck on something though, please, 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 please ask.